All right, great. Thank you, Professor Yu, for that very kind introduction, and thank you for the invitation to kick off this symposium and to be part of Future Science Prize Week. It's a very exciting event, and I very much appreciate it. So what I'm going to tell you about is three major transitions in microbiome research. The first integrating ecology, the second integrating machine learning, and the third that's going on right now, integrating AI. I have some disclosures, none are particularly relevant to this talk, except I used AI extensively for the illustrations. So why do we study complex microbiomes? Well, we study them because they're intimately involved in many of Earth's problems, and the difference between a good future and a bad future depends on how we respond to these different topics like infectious disease, chronic disease, food production, pollution, and climate change. And the tools to understand and apply these microbiomes are fundamentally grounded in ecology, although, as I'll show you more recently, applying machine learning and artificial intelligence has led us to a lot of advances that we could not do with ecology alone. Now, I did my graduate studies at Princeton in the Department of Ecology and Evolution, and one of the projects that I had was snorkeling around Panama, holding one of these 1 meter quadrats and recording all of the organisms that I saw in each of those square meters, looking at transitions across ecological boundaries. And what I learned there is that ecological sampling, no matter how you do it, is inherently biased. So, for example, anything that crawled out of my little frame, I would not be able to sample. And the taxonomic resolution is limited. You can't be an expert on everything that you look at. And to bring together specialists in hundreds of different taxa is essentially impossible. But despite these limitations in what you can do with ecology, no one particularly cares about them in that field, because if you have standardized methods that you apply to many places, you can make comparisons across sites, across time, across space, and derive a lot of useful information despite these biases. So the strategy of applying community ecology has been really transformative. We took techniques primarily derived from Whitaker's studies of landscapes in the 1960s, and so he was applying this mainly to forest ecology. And so there are concepts like alpha diversity, where you're looking at the number of different trees in one patch, or as I think you will see during the rest of the talks in this session, the number of microbes in one biological specimen. And then there's beta diversity, where you want to compare two patches to one another, and you're looking at the dissimilarity in the species assemblages between those two patches. And if you do many of those pairwise comparisons, you can build them up into a general picture of the similarities and differences among organisms in many samples. And these concepts of alpha and beta diversity really dominated microbiome research for a decade, from about 2005 to 2015. We were able to dramatically improve the rate of acquisition of the ecology data we needed to find out about complex microbiomes in 2008, when Micah Hamady, a very talented computer science grad student in my lab at Boulder, figured out that we could use formal error-correcting codes, such as Hamming and Golay codes, to tag the DNA that we amplified out of many different samples, and then mix the results from hundreds of samples together and sequence them at once on an expensive sequencing run from a sequencing instrument. And what we were looking at was one marker gene, the small subunit ribosomal RNA, which you can think of as being like a name tag for your bacteria. It's very phylogenetically conserved, so you can read out from many organisms what this particular sequence is and match it up to a database to find out who's there. And so in this one study, we looked at samples from cystic fibrosis patients' lungs, so an infectious disease example. We looked at rivers across North America. We looked at the air, an incredibly challenging low biomass environment that later became really important to study during the COVID pandemic. And then the Guerrero Negro microbial mat, the most complex microbiome we know of on Earth, although ironically NASA studied it as a model of simplicity because they thought the green things you see there, the cyanobacteria, was all that was in there, not the hundreds of other bacterial phyla. And so we collected 500,000 sequences on this run, and what you can see is that the sample types cluster beautifully together. we got a lot of information about each of these sample types that I won't go into, because what I want to focus on is the cost. So the 500,000 sequences we collected were about equivalent to the total number of those sequences that had been collected by the entire scientific community in the 20 years prior. And the Sanger facility down the hall from my lab at Boulder still charged $8 a sequence. So if we'd done it the old way, it would have cost $4 million and taken a decade. But with the then new 454 sequencing, it cost just $12,000, and we did it overnight. And so this approach was what opened up the Human Microbiome Project, the Earth Microbiome Project, the American Gut Project, which I'll talk about later, and many other large-scale sequencing efforts. But what's most amazing is that that amount of sequencing, it cost $4 million in 2007. We brought down to $12,000 in 2008. Today it costs you just 75 cents. And that's amazing, right? That's how we can look at so many samples and so many experimental designs. The decline in the cost of sequencing makes this stuff feasible to apply at scale. And so in the Human Microbiome Project, we got the first overall map of how human microbiomes are arranged using a toolkit called Chien, developed by Greg Cacapareso, then a postdoc in my lab, now a full professor at Northern Arizona University, using the unifrac distance metric introduced by Kathy Lozapone when she was a PhD student in my lab, and she's now a full professor at the University of Colorado. And so you can see almost emerging like continents on this map, the oral, the skin, the vaginal, and the fecal microbiomes, which are very different from one another. So you want to think of yourself not having a microbiome, but having a series of connected microbiomes across your body, and we're just starting to understand how they connect and change. Now, focusing in on the gut microbiome, this ecological approach where essentially you find for groups of samples, do they differ in their alpha diversity, and do they differ in their beta diversity, their community dissimilarity, led us to associate the microbiome with a whole lot of different processes, including its role in digestion, in the immune system maturation, in clearing pathogens, and even in things like brain development and behavior. But we're also increasingly finding it has a lot of negative effects. So when the microbiome goes wrong, it can lead to metabolic syndrome like diabetes, obesity, and so on, GI disorders, cancer, which you're going to be hearing more about in this session, and even neurological and neurodevelopmental conditions ranging from autism all the way to Alzheimer's. So the community ecology approach made a lot of really remarkable discoveries, including things like identifying the natural variation in the human microbiome, weather conditions, including obesity and IBD, two of the early ones we studied, decreased microbial diversity from a healthy state, and things like other microbial communities different on average between healthy and diseased states. So therefore, might the microbiome have a lot to do with things that no one had any idea it was associated with. But the problem comes when you have ecologists interacting with clinicians, where once you start to get into medical topics, you get questions like, what do all those dots mean on those principal coordinates plots? What are the axes? How do you link that to some kind of medical reality that you understand? Did you find a particular pathogen for real, which is really difficult with the kinds of amplicon sequencing approaches I told you about that are relatively low resolution? Which species are really making the difference, which again you can't do if you don't have species resolution on your underlying data. And then the real challenge, is this a clinical test? And basically everything I showed you there is research. And so community ecology using short-read data fundamentally can't answer these types of questions, although you can make a lot of really exciting scientific discoveries. So this led us in 2011 to explore the machine learning approach. And today we can build machine learning models that classify people or animals for a lot of different traits from their microbiome data. So for example, Dan Knights, who was a PhD student in my lab at the time and is now a full professor at the University of Minnesota, found that we could use random forest classifiers of microbiome data to classify an individual as lean or obese with 90% accuracy based on their microbial genes. Whereas if you do that from human genes, you can only do it with about 57% accuracy, even if you look at every mutation that's ever been linked to obesity by genome-wide association studies. Other researchers have shown that the microbiome affects the personalized impact of a huge number of drugs. So for obesity, you don't really need to sequence your microbiome to tell if you're fat or not. But if you want to understand, will you respond to a particular drug, that's invisible, and you really need to do the test to find it out. And if you do that test for painkillers like acetaminophen, for cardiac drugs like digoxin, anti-cancer drugs ranging from cyclophosphamide to the latest checkpoint inhibitors, whether they're safe, whether you'll respond, depends on the genes in those microbes in your gut. And we're now starting to wonder whether most molecules we ingest, whether they're drugs or food, whether the microbiome has a big impact on how our bodies respond to them. And this was taken to a really remarkable level by Aran Segal at the Weizmann Institute, who in 2014 published this groundbreaking paper in Cell, where he hooked 800 people up to continuous glucose monitors and then fed them a defined sequence of diets, and showed that how you individually release blood sugar after a meal is really different one person to another, even for the same foods. And so he measured a whole bunch of stuff for 800 people, including blood tests, questionnaires, body measurements, food diaries, and of course the microbiome. And one of the key findings from this paper was how different people are. So for many people in the study, and for many of you, you'd probably think of rice as a pretty healthy meal, right? But it turns out that for a substantial fraction of the population, it's worse for you to eat rice than it is for you to eat ice cream. And out of all of the things that they measured that were the deciding factor, essentially it was the particular microbes that people have. So on learning this, people typically have two questions. The first is there some sort of test I could do that tells me if I'm in the rice category or the ice cream category? So far, this only works for research in limited populations, but doing that for everyone would be really exciting. And then perhaps more importantly, suppose I do the test and I find out that I'm in the rice category. Could I change my microbiome to get over into the ice cream category? Which might sound frivolous, but in the case of drugs, where you want to find out if you're a non-responder to a drug and move yourself over into the responder category, that's deadly serious. So we're scaling this up in a project called Nutrition for Precision Health, or NPH, where one of the key parts of this is to develop algorithms that predict individual responses to food and dietary patterns. And this is Jack Gilbert, the other PI of the Microbiome Center that we lead at UC San Diego, in a sizable chunk of this $170 million investment from the Common Fund. And essentially the study design is to do 10,000 people cross-sectionally, and then longitudinal studies of 2,500 people at home, and then 500 people in an inpatient setting, to really get to the bottom across many different biomarkers of what's most important to predict your response to individual foods, so we can produce better guidelines for the whole population. So machine learning can tell you a lot that you can't get at with ecology. For example, the classifier accuracy scores can tell you how accurately I can classify a single sample as being healthy or diseased. The feature selection algorithms usually tell you which particular features are most important for that classification, so you know what microbes are involved. And then you can tell if the classifier is robust and reproducible. So for example, in IBD, we were able to train classifiers on US populations of patients and apply them well in China. We were able to apply them all the way from humans to dogs. So you can get these very cross-generalizable classifiers that you can apply even across the species boundary. However, again, when you get to the interaction with clinicians, you run into trouble. So, for example, they have a lot of questions like, what does classifier accuracy mean? What are all these acronyms that you have to learn about the machine learning stuff to be able to explain it to yourself or to patients? You want to know whether the features are causal, and essentially anything you can get, random forest, support vector machines, artificial neural nets, and so on, are association, not causality. Again, you want to know what species are making the difference, which you cannot get at if you don't have species-level resolution input, which you typically don't with short-read sequencing methods, which are prevalent in the field. And then finally, there is a problem of how you explain this to patients, especially if it was a long time since you took calculus and you really don't want to explain different gradient descent algorithms or what a Jacobian is or any of that kind of stuff. So machine learning is still hard. Typically, to do it right, you need a large multidisciplinary team of people to work together across disciplinary boundaries, and it still can't answer these key questions that you need in a clinical setting. So this is where we're turning to AI, and I know everyone says that they're doing AI now, and you're probably sick of hearing about it. We launched the Artificial Intelligence for a Healthy Living Centre as a joint project between UCSD and IBM back in 2017 that I co-directed for the first 6 years of its existence. And this was part of the AI Horizons network from IBM, initially focusing on the Watson technology and then rapidly pivoting to other areas of AI. A lot of what I've told you about so far is conclusions that you draw on large populations, but we really want to be able to take this to the individual level. And the individual I'm going to talk about is possibly the least private person on Earth, so none of this violates medical privacy rules. It's my colleague Larry Smarr at UCSD, who's a physicist who got interested in the complex system of his own microbiome. And he's a very interesting individual because he's been releasing data about himself on the internet for the last 25 years, starting with one dimension, which was his weight over time, and then going to dozens of dimensions with blood tests, thousands of dimensions with human genetic profiling, and now millions of dimensions with the microbiome. And shortly after I got to UCSD in 2014, I had a meeting with Larry where I was explaining to him the Human Microbiome Project data set, much as I showed it to you earlier in the talk, in front of a 64 million pixel display wall. So this is a live rotatable image in 64 million pixels. And he said, well, you know, Rob, I have a very interesting microbiome myself because I'm an IBD patient. How do I get myself onto that map that you made of the microbiome? So shortly after that, this box showed up in my lab, the first one of many. Larry's still on dry ice. I told you he was an interesting character. And he'd actually been doing this for a while. So when we reprocessed all his data, this is what he'd been looking at. And this is a family level of different kinds of microbes in his gut shown with different colors over a period of 3 years. And don't worry if you don't see anything useful in that, because neither did Larry, despite spending some giant amount on sequencing and personnel effort to get to this point. So we reprocessed the data with our tools. In technical terms, this is a principal coordinates plot of unifrac distances between samples. And what you can see, this is just rotating the data frame. I'm going to animate it, and you'll see that he starts at one edge of the blue, and then his successive time points have him traveling through the blue region, crossing over into the red region, and then he bounces around basically at random in the red region. So what, you say? Well, what we can do is we can couple that to all that medical data Larry collected about himself. And what we can see is that this initial shift was caused by a course of antibiotics. This directional travel through the blue configuration space is when he's having frequent IBD symptoms and losing weight, and his life isn't very good. The switch is caused by a change in medications. And then for that region where he's bouncing around apparently at random in the red, his IBD symptoms are basically gone, and he recovers his weight back up to a healthy set point where he stays for about a year. And this is amazing, right? If we've been able to tell Larry about these different regions of configuration space, we could have told him, you know, while you're in this blue region traveling directionally, you really need to do something about that as soon as possible. When you're in this red region, even though your microbiome configuration is not changing, it's in a stable configuration where your health will improve. But the state of the art then was we could only read it out retrospectively, and what we need to do is we need to be able to model it predictively. So that wasn't the end of Larry's health journey, so now I'm showing you a few more years of it, and a few events are big enough that you can see them even on that taxonomy plot. So for example, he had to have 20 centimeters of his sigmoid bowel removed in 2016 as an attempt to cure his IBD symptoms. At about the same time, we were running the American Gut Project, where we looked at tens of thousands of people, citizen scientists contributing samples, and crowdfunding the project to understand what had a large versus a small impact on the microbiome. And on these power curves, what you're looking at is the number of samples per group on the x-axis versus the p-value of your significance test at the extremes of those groups on the y-axis. And so basically, the steeper the curve, the bigger the effect. And for things like age and antibiotics and travel and even things like alcohol and exercise, you can see the effect with a few hundred people down to a few dozen people for the stronger ones like age. But the largest effect is just the number of different species of plants you ate in the week before you sent in your sample. And that's fascinating because that's something you have complete control over, unlike your age unlike disease markers that are not shown on this plot, but are at about the same level as salted snacks. And this turns out to also be true for the metabolome. So one thing we're doing a lot of at the moment is integrating the microbiome with direct chemical readouts of the gut with my collaborator Peter Dorrestein. And if you eat more plant diversity, you also have more diverse molecular features when we look at your stool or when we look at your blood sample. So Larry saw this result and he wondered, could he improve his microbiome diversity and therefore his health, not just by settling for 30 species a week, which was the top of his range, but by consuming 60 plant species per day. So he started making this green smoothie and drinking it every day. If you're interested in his actual ingredients, we have them on the MicroZeta website. So that's the umbrella project for the American Gut Project, including various international expansions. And this was also featured on the Emmy Award-winning Hack Your Health documentary, if you want a more entertaining view of this than I'm giving you today. However, I'll admit that the presenter of Hack Your Health had a pretty hard time understanding all those complex displays that I showed you, which are coming out of the ecological approach, and also had a pretty hard time understanding why anyone would actually want to consume the smoothie, given the ingredients that were in it. And having tried it, I have to say that I don't recommend it from a taste perspective. I only tried it once, and that was enough. But what it did have a big impact on was his blood glucose. So he'd been slipping from the normal range. So that line of 100 is basically the boundary between normal blood glucose and prediabetes. And so he'd been slipping into prediabetes and this transition into metabolic syndrome over the course of about 3 years. And the last 3 months of this, he had been eating vegan and gluten-free in an effort to reverse it. And you can see that it did absolutely nothing. But the green smoothies had a huge effect and put him right back down into the normal range for fasting glucose pretty much immediately and durably. And this was also coupled to a big decrease in his total fat, which he measured on his $100 Wi-Fi scale at home. He thought that result was too good to be true, so he went into the clinic and stood on a $17,000 scale, which confirmed the result, looking at a 36% drop in visceral fat specifically. So very large impact of this of introducing this plant diversity to his diet. And fascinatingly, most diet-based interventions, as well as most probiotics, don't change your microbiome enough that you look like a different person, because the differences in the microbiome between different people are really large. So your microbiome is probably 90% different from the microbiome of the person you're sitting next to. But Larry was actually able to become 5 different people over 8 years in terms of his microbiome configuration, especially with the surgery, which had a huge effect, the vegan gluten-free period, which had a big effect on his microbiome, but not on his health indicators, and then the green smoothie time-restricted feeding period in green here, which did have a big impact on him metabolically. So what we're trying to do is we're trying to take all of Larry's rich data and build a digital twin of him. And for manufactured products, digital twins are becoming the new normal. So, for example, Porsche has a digital twin of every car they sell that predicts that components will fail before they do, so you can do preventive maintenance instead of having to call for rescue on the Autobahn or whatever. And this kind of thing has been applied to biological systems. The first attempt to do so was by Marcus Kovitz's lab back in 2012, looking at Mycoplasma genitalium, a very simple microbe with just 525 genes. But what you can do now is you can extend it to the whole microbiome using a technique called flux balance analysis on whole genome reconstructions. And so we've been doing some pilots on this with Larry, together with Enos Thiele at the University of Galway in Ireland. And the first pilot, looking at just 7 stool samples, what we saw was that some metabolites were 10 times to 10,000 times overproduced in Larry versus a healthy reference population. But we are now doing it with hundreds of samples that he's collected over the period of 15 years and doing it with much higher resolution methods to be able to give them a much better basis for the modeling. So right now, the digital twin we can make of Larry is pretty bad, right? It's crude, it's pixelated, it's low resolution, it's decontextualized from its environment. But what we want to be able to do, and we're doing this with a combination of long-read sequencing, transformer models, as well as flux balance models, and spatial omics techniques, we want a much higher resolution model that can contextualize them in different places, run counterfactual scenarios, and get accurate representations of his reactions. And so the big breakthrough here, performed by Omar Din in my lab shown here, and Jake Minnick, who should be showing up here but isn't, a former grad student of mine who just started a faculty position at Baylor. What we've moved to is a long-read metagenomics approach, where instead of small fragments that span just 10% of a genome when we try to assemble them, you can get dozens of complete circular closed bacterial genomes from every sample. So we have a much better ability to read out the genome of the species and even the strain level that you need to make clinical decisions than has ever existed before. And this came out in Cell a couple of months ago. But the real problem is that even if you can read out a huge number of genomes at high resolution, still understanding all those thousands of Latin and Greek names is really hard. And the real problem we're trying to tackle is to build a user interface for our microbiomes. And what you want to be able to do is when you're standing there in the supermarket confronted with 1,000 kinds of yogurt, is to be able to look at one with your phone and ask your phone in natural language questions about it. So I'm sure you've all used ChatGPT by now. We're calling this something related but a little different given the application domain we're targeting. But the idea is you should just be able to talk to your phone and ask it, should I eat this product I'm scanning to improve my health? And then if the answer is no, maybe you can use the phone's camera to highlight with augmented reality the other products that are on the shelf that you should eat instead of the one that you're considering. And what we'd ideally like to do is deploy this kind of thing everywhere. So the Gates Foundation has discovered that the best way to support child health is to support maternal health so that babies in the worst risk categories are born into healthier risk categories from the beginning. So you could imagine deploying this sort of thing in Bangladesh, for example, so that an expecting mother could ask, should I eat this fruit or vegetable or that one so that my baby will be healthy? And that might sound absurdly ambitious, but smartphone penetration in India is already up above 60%, and it's projected to be 95% by 2040. Bangladesh is only about 10 percentage points behind. So the idea that we can develop this sort of thing in high-resource settings like the United States and Hong Kong and Singapore, and then deploy it globally within the next 20 years is really exciting. So that's the kind of crazy stuff we're up to at the Center for Microbiome Innovation that I direct at UC San Diego, where together with over 140 faculty members, we're targeting a lot of hard problems like this in human health, in agriculture, in ocean health, and in all kinds of other things where the microbiome is involved. And so the really exciting prospect is that AI, unlike the previous generations of tools, may finally let us deliver what physicians really want and need in terms of translation and upscaling different kinds of microbiome data, including the old low-resolution data sets, getting explainable and accurate and personalized predictions, incorporating LLMs to make them friendly. And then I didn't talk about this here, but we're very excited about generative AI to design new microbiomes, new diets, new therapies from scratch from the ingredients with sufficiently large training sets. And then the natural language interaction we think is going to be really key, especially being able to ask for explanation of microbiome results trained on the trustworthy scientific literature rather than all of the internet. So together these advances will transform our ability to not just know about our microbiomes, but to actually use them. So with that, I'd like to thank the amazing people in my lab, in Peter Dorrestein's lab, my metabolomics collaborator, and the Center for Microbiome Innovation. And I would like to thank you all. Thanks again for the invitation to start off the session at the Symposium, which I'm very much enjoying. And I look forward to your questions in the panel discussion. Thank you.